We've got the chloroplast. So this structure here represents the chloroplast. If we're talking about photosynthesis, if you recall from our content over cell organelles, we know that photosynthesis is going to occur in the chloroplast. With our equation present, what we do know is that it requires carbon dioxide, water, and anytime you need energy for a chemical reaction to take place, you're going to write the type of energy right over the arrow to indicate that energy was required. So we do need energy that we're going to get from the sun, and I say we, I'm talking about plants here, to produce or to yield glucose, which is the main point of photosynthesis. That is the purpose, if you will, of photosynthesis, is to create glucose. We're also going to end up creating some oxygen as well. Within the chloroplast, you have several structures. A few of these should look familiar to you. And despite not having enough room to label everything inside the chloroplast, I would highly recommend that you create a little key for yourself somewhere, either on the page where your notes are or in your journal. I did something very similar because you're going to see some things that hopefully you recognize. So first off, right here, these should look familiar from our coverage of the mitochondria and cellular respiration. These look like ATP synthase because they are ATP synthase. So one thing you can do, because we don't want to take up space within here for our little processes that we're going to be observing, is maybe somewhere off to the side, like I said, create a little key. So I'll draw ATP synthase right here at the bottom and label it ATP synthase. This is what I did in my notes. I would recommend that you do it in yours as well. Just so you can keep track of each of these different structures. I've tried to give them a distinct shape as well as incorporate some color or some diagonal lines in there to tell them apart. So first off is ATP synthase. We are already familiar with what that does. We know that it makes ATP. So within the chloroplast, there is a process for creating ATP, and we're going to talk about what the use of it is and obviously how it's going to work. We've got these other structures here that kind of resemble salt and pepper shakers. These little dots here at the top should be green. Your copy is probably not colored, but I would indicate that those are green. And these are going to be what we call photosystems. So we'll put our little dots there. Like I said, kind of looks to me like a salt or pepper shaker. And that is going to be a photosystem. And we have two of them. Right next to or right in between our photosystems, we have something that looks like something we've seen before, and that's going to be our proton pump. If we're going to have ATP synthase, we're probably going to need a proton pump, if you recall from cellular respiration, to make sure that we have the right concentration of protons so that they can flow in the direction that's going to help ATP synthase do its job. All right, and then last but not least, in this structure here, we have this little box with its diagonal lines. I'll put this one down here at the bottom. There's my diagonal lines. Now, it doesn't normally have diagonal lines. I'm just trying to set it apart from our proton pump. And that is going to be referred to as our docking station. Now, it does have a name, a more scientific and appropriate name for the structure. However, for our purposes, we only need to know it as the docking station. So we're going to refer to it as the docking station, which should make some sense here once we start actually looking at the process. And then this entire structure here is what's known as a, and I'm not going to draw this again, but this entire structure here is known as a thylakoid. All right, so this whole thing here is a thylakoid. Now the thylakoids, when you look at chloroplasts, and don't draw this in the middle of your chloroplast, I'm going to just because I don't have anywhere else to do it. But when you look at a diagram of a chloroplast, excuse me, please excuse my dog scratching itself or shaking in the other room. If you have a chloroplast and we did a cross section of it, what you're going to notice on the inside are these green discs, kind of these flattened green discs. And a lot of people refer to these as the green pancakes or stack of coins, whatever you want to use to indicate what they are. But inside of a chloroplast, you're going to notice that you have these structures that look like stacks of pancakes that are green. They're all interconnected and they fill most of the space within the chloroplast. That is what this represents. It represents one of these little flattened discs. These are thylakoids in stacks. So this represents one. Your diagram has two to kind of show you that they are stacked. Now the thylakoids obviously have a lot of structures in them. We're going to talk about their job here specifically. But the reason for the stack is to increase surface area. Very similar to how the mitochondria's inner membrane is folded to increase surface area. We stack these thylakoids in such a way to increase the amount of surface area we have as well. Kind of like when you look at a journal. So this is 70 sheets. Each one has a certain amount of surface area. If we use numbers that are easy to work with, let's say this is 12 by 12, which it's not, but let's just say it's 12 by 12. That would mean that I would have 70, if I just use the front, or if I use front and back, 140 square feet of surface area within this journal. Now, the reason it doesn't take up too much space is because we stacked all the pages. If I laid these out on the ground, they'd be bigger than this office. So we're minimizing the amount of space required to house a great amount of surface area by stacking these up. Like I said, very similar to how the mitochondria has that inner membrane that's folded to increase surface area. All right, so looking at what we've got, we've identified our structures. This is a thylakoid. These are our photosystems, our proton pump, our docking station, and obviously our ATP synthase. The sun's going to play a part here. And then we have our Calvin cycle. Now the Calvin cycle, it's not really the same as the Krebs cycle, but it is a cycle within the organelle. So it has one just like the mitochondria does. And this one's going to be responsible for putting things together for us. And we're going to talk about that here in just a second. But when you look at the diagram, it's kind of separated into two sections, right? We could probably divide it in half, and one side would be thylakoid, the other side would be Calvin cycle. And the reason for that is to indicate that one side is light dependent, while the other side is light independent. Thylakoids require sunlight to function. To do their job, they require sunlight energy. The Calvin cycle does not require sunlight. This would run and this would work at night without any sunlight whatsoever. Now, it's not going to be able to do its job unless the thylakoid is doing its job. So you could say that indirectly it needs sunlight, but it does not directly depend on sunlight to carry out its processes. And the thing about the Calvin cycle is that it's a series of chemical reactions. I believe 9 different chemical reactions that take place to help us eventually make glucose, which, as I mentioned, is the sole purpose of what we're trying to do here. All right, so if our goal is to make glucose, which is C6H12O6, let's write that over here off to the side. C6H12O6. This is our goal. We want to make glucose. And if you want to make something, it helps to know what you need to make it. So if we consider this the recipe, right? We're trying to figure out the recipe for glucose. There are 3 things that we're going to need. The first thing we're going to need are the raw materials: carbon, hydrogen, and oxygen. The C, the H, and the O in glucose. All right, that's the first thing we need, these raw ingredients. Secondly, we're going to need electrons. We usually write an E with a negative symbol to indicate electrons. And then third, we're going to need ATP. Now, the reason for all of these, well, at least the reason for the first one's pretty obvious. We need carbon, hydrogen, and oxygen because glucose has carbon, hydrogen, and oxygen. We need electrons because electrons are responsible for bonding. We know that when 2 atoms bond together to create a molecule, they bond because of their electrons. So we're going to need electrons to kind of put everything together and make sure that it holds together. ATP is going to be required because we're trying to take something that is not organized, something that's, parts of this glucose, and put it together, but more importantly, put it together in the right shape. Anytime you try to create order out of chaos, this is like the opposite of entropy. If you're trying to create order out of chaos, it's going to require energy. And if we want glucose, we can't just put carbon, hydrogen, oxygen together in any form. We need to make sure it's the right shape. To make sure it's the right shape, we're going to have to use some energy. And the cell likes to use ATP for energy, so we're going to use ATP as well. All right, so let's talk about where we're going to get these ingredients or these materials by starting with the one that we're probably most familiar with, and that's going to be ATP synthase. Now, we know that ATP synthase, if we recall our cellular respiration notes, has 2 qualities that make it unique and viable for creating ATP. 1, it's hollow, and 2, it spins. Now, because it's hollow, we're going to allow, or it's going to allow, protons to pass through it. And we want protons to pass through ATP synthase because when protons pass through ATP synthase, it spins, right? So it's hollow, which allows the protons to travel through the thylakoid out into the, we haven't identified this part. This is not called the matrix. This is going to be called the stroma. So when protons flow from the thylakoid out into the stroma, ATP synthase will spin. When it spins, it's able to take ADP and a phosphate and kind of force them together into a bond to make ATP, using the expo as an example like I did last time. So we want to maintain a higher level of concentration on the inside of the thylakoid than we do on the outside in the stroma, so that through passive transport, more like facilitated diffusion, right, we can get protons moving through ATP synthase. Now, the way this is going to look, if we're trying to map out what's happening within the chloroplast, here we have ADP. If this will work, there we go. Here we have ADP and phosphates that are getting picked up by ATP synthase, turning into ATP because it's spinning around and turning them into ATP. And that ATP is going to be sent to the Calvin cycle. So ADP and the phosphate picked up by ATP synthase, which is spinning because protons are going through it, turns into ATP, which we're going to send to the Calvin cycle. And that already takes care of one item on our list. All right, 3 ingredients, ATP is one of them. We create it with ATP synthase, no different than what we did in cellular respiration. However, be clear on this. This ATP is just for photosynthesis. This ATP goes through the Calvin cycle to make glucose. This is not ATP that's getting sent to any other organelle or any other parts of the cell for energy. Plants have mitochondria. Mitochondria is going to be responsible for cellular respiration, that is going to provide the ATP for the rest of the cells, right? So that it can continue to live and carry out all of its functions. This ATP is just for photosynthesis, specifically for the Calvin cycle. All right. Now, if you recall, we want to maintain a higher level of, or a higher concentration of protons inside the thylakoid. And if we keep having them flow through ATP synthase, eventually we're going to reach equilibrium. To prevent that from happening, we have a proton pump. We've already identified it. And we know that proton pumps are going to pump protons into the thylakoid. That is their job, to pump protons back into the thylakoid to make sure we have a higher concentration on the inside so they can flow out of the thylakoid into the stroma. And keep in mind, this is happening in all of the thylakoids that are stacked up throughout the chloroplast. Now, if you remember, if it has the word pump, that means it requires energy. It can't be passive transport like our facilitated diffusion down here, because we're trying to go against the concentration gradient. As such, we need energy. Where are we going to get that energy? Well, if you think back to cellular respiration, we got our energy from electrons. We were breaking down glucose into pyruvate. Krebs cycle was stealing electrons via these electron carriers and sending them down to our electron transport chain, which was part of our proton pumps, right? And it provided that energy. We're going to use electrons as well. However, it's going to be a little bit different. So the photosystems here are going to get some light energy from the sun, right? We're going to get some light energy from the sun. The chlorophyll inside the photosystem, that's what the green dots represent, has electrons. When you add energy to an electron or to an atom in general, it's going to cause the electrons to go up a level. Okay? As they go up a level, they're energized, they're not stable, and they'll fall back down a level. When they fall back down, they release the energy that they had stored in them that they got from the sun. This is why when you put something dark outside, it gets hot. It absorbs energy from the sun. Those electrons can't stay at that high level. They'll drop back down, releasing the energy in the form of heat. So these electrons get energized. Chlorophyll has this unique way of holding on to the electrons so it doesn't drop back down immediately. So it transports it to our pump where it drops a level, releasing its energy so that it can power the pump. The electrons that no longer are energized get transported to the second photosystem here, where sunlight will energize them again and pass those on to our docking station, okay? So we're still powering our proton pump with electrons. We're just using electrons from chlorophyll, okay, and energy from the sun to give them the energy they need so that the proton pump can have the power that it needs to pump protons back into the thylakoid. We energize them again because we're sending them to this docking station. And the reason for that is because we need electrons for photosynthesis. We need electrons to make glucose. And we're going to put everything together in the Calvin cycle. So we need to get electrons to the Calvin cycle, similar to how we got ATP to the Calvin cycle. This docking station is holding on to these energized electrons for us. So what we're going to need is an electron carrier. And we had electron carriers in the Krebs cycle for cellular respiration. Our electron carrier in this case is NADP plus. In cellular respiration, it was NAD plus. So we're adding a P in there for photosynthesis. That's not what it stands for, but that's one way to help you remember that this is NADP plus. So that's an electron carrier. It's going to come around to the docking station, pick up those electrons, and then it'll turn into NADPH. And NADPH will send the electrons to the Calvin cycle, which checks off a second item from our list of ingredients for making glucose. All right, so quick recap. The thylakoid, which has all of these structures in it, ATP synthase, photosystems, proton pump, and docking station, has protons inside. We're maintaining a higher level of concentration inside so they can flow through ATP synthase, which is hollow. When they flow through, it spins. When it spins, it takes ADP and a phosphate, combines them to make ATP, which we're going to send to the Calvin cycle. The photosystem will get energized with energy from the sun to raise those electrons up a level, transport those to our proton pump where they'll drop down their level, releasing their energy to power the pump to get protons back into the thylakoid to make sure we have a higher concentration. Those electrons get transported to our second photosystem here where they get energized again, and then transported to the docking station where NADP plus picks them up and carries them to the Calvin cycle. That takes care of our energy and our electrons. That leaves us with the raw materials. Where are we going to get our raw materials? Carbon, hydrogen, and oxygen. Now, before we get into that, I do want to make note of one thing. This set of structures right here, if you paid attention to the way I was phrasing this, the electrons are being transported to each structure along this chain, if you will. So this is also an electron transport chain. Okay? It doesn't look the same as the electron transport chain that we had in cellular respiration in the mitochondria, but it is still a type of electron transport chain because electrons are being transported from one structure to the next down this chain, if you will. So this is also an electron transport chain. Just want to throw that in there as an aside. All right, so let's talk about where we're going to get our raw materials. Where do we get our carbon, our hydrogen, and our oxygen? Well, we're going to get our carbon, hydrogen, and oxygen, well, specifically carbon and oxygen from, I'm going to try not to knock over my tripod here, from the air. So here's the air, and it's going to provide us with CO2. So we're going to get our raw materials, our carbon and oxygen, directly from the air in the form of CO2. And we're going to say 3 CO2 so that we can understand how this Calvin cycle works. And it helps if we can keep track of one of these elements, and we're going to keep track of carbon. Very similar to what we did with cellular respiration. All right, so we get carbon and oxygen from the air in the form of CO2. We're going to get our hydrogen in the form of water. We get hydrogen in the form of water. Also, we're going to get a little bit of oxygen in there as well, but that is not our main source. It is, however, our main source of hydrogen for photosynthesis. Now, water is going to be transported via the root system, right? This is how it's going to get water. It'll transport it from the roots up through the xylem, the vascular tissue of the stem, and get it to where it needs to go within the plant. Most of this is going to be happening within the leaves of the plant because they're the ones that are going to have the most amount of direct sunlight based on their shape. Okay, so a lot of this needs to get sent to the leaves. The leaves have special structures that allow them to exchange gases with the environment, where they're able to get CO2 from the air and release the excess oxygen that they create. But we'll talk about that in just a bit. All right, so water, we've already talked about some of its unique properties, but it has a property that we have not talked about, and that is its ability to disassociate, where it can break down into hydrogen, H+, and OH negative. Now you'll notice we still have what we need to be water, right? We still have 2 hydrogens and an oxygen. But this is one of those things about water that kind of puts it in the middle between acids and bases, and it is a universal solvent. But as such, it can dissociate on its own. It can separate into H plus and OH negative. These H pluses are where we're going to get the hydrogen that we need for the Calvin cycle. So at this point, we have everything we need to make glucose. Now, within the Calvin cycle, we have, I believe, 9 different chemical reactions that are going to take place to create glucose. We don't need to know all of them. All we need to know is that we get our raw materials from the air, right, from the environment, from water, and from the thylakoids. That's where we get all the materials we need. The Calvin cycle is going to put them all together. The example that I've been using with most students is that if I wanted to make a pizza, I'm going to need dough, I'm going to need sauce, I'm going to need cheese, and then any other toppings, right? If I took dough and sauce and cheese and the toppings and I just put them all in a container, I have all the ingredients, but it's not a pizza yet. Most people are aware that pizza has a certain shape to it, right? They specifically look for a level of organization that you're not going to find if you just put all the ingredients in a bag or a container. So here's where we're getting ingredients, right? The thylakoid, the air, the water. We're going to put them all together in the Calvin cycle where it's going to organize it into what we want it to be, right? It's going to put that dough, then it's going to put the sauce, then it's going to put the cheese, then it's going to put the toppings. It's going to organize it so that it is what we want it to be. And in this case, we want to take all of these ingredients and make glucose, very specific molecule. So what we're going to notice as it goes through the cycle, if we follow 3 carbons from CO2, is that we're going to end up creating a molecule called, well, it's not called G3P, but that's what we're going to call it. It has its own name. It's a complicated name, and you don't need to worry about it. We're just going to say it's G3P, and it is half a glucose. Now, I know what some of you are thinking, isn't half a glucose pyruvate? Yes, in cellular respiration, before we get into the mitochondria, we undergo glycolysis where we split a glucose in half and end up with 2 pyruvates. However, the shape of the molecule plays a part in what it is. So just because we have half the components for glucose does not guarantee we're going to have pyruvate. And in this case, through the Calvin cycle, we create G3P instead, which still has half of the molecules or half of the atoms we need for glucose. So different molecule, still half a glucose. Now, if the goal is to make an entire glucose molecule, we can't just do 1 complete cycle. We're going to have to do 2. So after 2 cycles, we end up with 2 halves of a glucose, which will result in 1 glucose. Right, our C6H12O6, which makes sense. We had 3 carbons going through once. We're going to get half of the carbons we need for glucose, and we need 6. We got 3. We do it twice. We end up with 6, and we end up doubling what we needed for our hydrogen and oxygen as well. And essentially, that is photosynthesis. We get our electrons and our ATP from the thylakoid. We get our carbon and oxygen from the air in the form of CO2. We get our hydrogen in the form of water from water. And the Calvin cycle puts it all together to create glucose. That is photosynthesis. However, when we looked at cellular respiration, we started from the end and worked our way to the beginning, and we started addressing problems to determine where we were getting everything that we needed, right? If we run out of protons, we can't have ATP synthase doing its job, so we got to figure out how to get protons back into the inner membrane. So we're looking at some of those things here already, but there is an issue that we have not addressed yet that we need to address, and that is sunlight does not provide us with electrons. Sunlight provides us with the energy to energize electrons. And if I'm using the electrons that are in the photosystem in chlorophyll and transporting them down the chain to the docking station, and NADP plus is picking up those electrons and sending them to the Calvin cycle to help us bond together our raw ingredients over here to make glucose, eventually we're going to run out of electrons. And we have not addressed that issue yet. So we need to talk about where do we get electrons so that we don't run out. Now keep in mind, plants have to do this to make glucose so that they can undergo cellular respiration. We already know that if you can't undergo cellular respiration, if you're not making ATP, you die. So they need to make sure they're making enough glucose so that they can make ATP, just like we do. The benefit to them, obviously, is they don't have to eat. They can make their own glucose. So we don't want to run out of electrons, because if we run out of electrons, we can't make glucose. If we can't make glucose, we can't undergo cellular respiration. which means we can't make ATP and we die, we being the plants, right? So let's talk about where we get our electrons. We're going to get electrons from water. Now we need to go back to something we've already talked about in cellular respiration for this to make sense. So bear with me. If you recall, in cellular respiration, we breathe in O2 and we eat food, right? So that we can make ATP. Now the whole reason we breathe in O2 is because O2 has formed, oxygen has formed covalent bonds with oxygen to be stable. Remember, you're stable if you have 8. So it has 6 valence electrons, so it'll bond with another oxygen and share 2 of them. This will make it stable. This is O2. We breathe that in. The main reason for doing this is so that we can take electrons away from pump 3, so that we can keep having electrons flowing down our electron transport chain in cellular respiration, so we can keep pumping protons into the IMS, so we can keep making ATP. So here's how we make this turn into water in cellular respiration. If you recall, when they pick up electrons from the third pump, right, from cellular respiration, if they pick up electrons from the third pump, it breaks the bond that holds them together. They each grab their own 2 extra electrons to be stable, right, which gives them a negative charge. Those negative charges are going to attract protons, because there's protons floating around in the matrix of the mitochondria. And anytime you have a proton and an electron, that makes hydrogen. So these oxygens that we breathed in to pick up electrons from the third pump grab onto the electrons, which attracts some protons, which turn into hydrogen. So now they both got 2 hydrogens, which makes this H2O and makes this H2O, right? So one way to look at hydrogen is it's just a proton and electron. Now, there's water in the chloroplast, there's water in the plant cell, and there's water in our thylakoid. So to indicate that, I'm gonna put a little blue dot in here that's gonna represent a water molecule. So just right here, that's a water molecule, okay? So here's the thing. Within the thylakoid, we need electrons. And at some point, we're going to run out. So we need a source of electrons. We're going to steal electrons from hydrogen because it has 1. So we're going to steal electrons from hydrogen that is bonded with our oxygens. And if I take away these electrons from hydrogen, I can have electrons for my electron transport chain, which means I have electrons that can go to the Calvin cycle. But if I take away the electrons from water, from the hydrogens that are attached to these oxygens, these protons have nothing to hold on to anymore. So these protons are going to end up just floating around the thylakoid, which is great because we want a lot of protons inside the thylakoid. So we're going to get extra protons from water. We're going to get electrons from water. And if we're going to be using these for ATP synthase, well, these are going to go away. And I'm going to be left with just these oxygens that are no longer stable. And when oxygen's not stable and it needs to bond with something, it'll bond with itself and become O2. So water within the thylakoid is going to provide us with electrons for our electron transport chain. It's going to provide us with protons for ATP synthase to use. And it's going to leave us with oxygens that bond together to create O2. This is the opposite of what happens in cellular respiration. And that's one way to look at this whole process. This is basically the opposite of cellular respiration. This is why we produce O2 in photosynthesis, because we're taking the electrons and the protons from hydrogen that have been bonded to these oxygens in the form of water to use for ATP synthase and our electron transport chain. It leaves us with these oxygens that become O2. The plant cell produces so much oxygen that even though it needs some of this for cellular respiration, because we know we need glucose and oxygen for cellular respiration, it produces so much through this process that it can afford to get rid of quite a bit of it, which is to our benefit, obviously. Within the leaves, we have these special little openings called stoma, not stroma, but stoma without the R. And the stoma have these little guard cells that keep them closed. And when it needs to, it opens them up. When it opens them up, they're able to get rid of excess oxygen and bring in the CO2 that they need. Remember, we're getting that from the air. So the leaves are opening and closing to exchange gas with the environment. This is a process called transpiration, not respiration, but transpiration. So they exchange oxygen for CO2 because they make so much of it, and whatever they need, they use obviously for cellular respiration. So obviously this works really well. If we look at both of them side by side, both of these processes are going to be occurring within a plant cell. The chloroplast makes the glucose, produces a little bit of oxygen that it needs. All the excess goes away into the environment, and then it sends it over to the mitochondria. The mitochondria picks up the glucose and the oxygen so it can go through the process of cellular respiration to create the ATP the rest of the cell needs. And that, all right there, is photosynthesis. So now at this point, I'm going to stop recording.